What is the chance of a female with blood type A and a male with blood type B to have a child with blood type O? Well, what we have to do here is make an assumption about what the parents could be. And the only way that this is possible for them to even have a baby with type O is if both of them are actually heterozygous. So the female would have to have an A and an O, and the male would have to have a B and an O. So we're going to make an assumption here that these are their genotypes. It's just the only way that it's possible. So we set up our Punnett square with the female and the male's blood type and fill it out. And what we're going to find here is that there's only one situation where they could have a child with blood type O. So that gives us a 25% chance of having blood type O if, and only if, parents are IA-IO and IB-IO for their blood type. Again, it's possible that they could not have a child with blood type O, but we're making an assumption here. A normal male and a female who is a carrier of hemophilia marry. What is the probability that a child produced by these two individuals would be born with the disease? Well, there's actually a really big hint here. This is a sex-linked trait, and we can see that because we're given X and Y and X in our problem. So knowing it's a sex-linked trait, we have to set up the Punnett square accordingly. So when we set up our square, we usually put the female at the top of the Punnett square just by convention and the male at the side of the square, and that's what we're going to do. So I put the female genotype at the top and the male genotype at the bottom. And when we cross and make our Punnett square here, what you're going to find is you end up with half females and half males. Remember, XX is a female and XY is a male. So now that we're going to look at the possibility of them having a child with hemophilia, we have to find someone who has either two little h's or one H and a Y, and there's only one spot where that occurs in this case. This boy only has a little h, and that means that he's going to have hemophilia. He has no normal gene, so this is a just 25% chance of having a child with the disease. And we know it's going to have to be a boy in this situation because each of the girls have at least one normal gene keeping them from having the disease. A man that is red-green colorblind has a child with a woman that does not carry the colorblind gene. What is the probability that a child born from this union will be colorblind? Well, again, we know that this is a sex-linked trait because of the X's and the Y's in the problem here. So we set up our Punnett square with the female's information on top and the male's information on the side, and we cross the genes. And what we see here is, well, everyone has at least one big N. So in this case, there's actually no chance of having a colorblind child. Now, the two girls do carry the gene, but they in fact have normal vision. Eye color is a sex-linked trait in Drosophila, which is a fruit fly. Red eyes are dominant over white eyes. Use a Punnett square to determine the possible genotypes and phenotypes of a cross between a white-eyed female and a red-eyed male. Well, we see that it's sex-linked, and that gives us a hint about what this is going to be like. So we know we're going to be crossing two X's from the female and an X and a Y from the male, and we've got to figure out what the gene is that they have on these chromosomes. Now it says that it is a white-eyed female and a red-eyed male, and that red eyes are dominant over white eyes. So in order to have a white-eyed female, she has to have both recessive genes, or little r's in this case. And the male has red eyes, so he's got to have a big R for red eyes because they're dominant. So we can set up our cross now and cross the flies. And what we see are half have a big R and a little r. And it's all the girls actually. And the other half, they also have the same genotype. They have a little r. So our boys have one genotype and our girls have one genotype. So when we write this out, we see that there's a 1-to-1 ratio of X big R, X little r, to X little r, Y. And in our phenotypic ratio, what this means is that there's one red-eyed female for every one white-eyed male. Straight wings are dominant over curly wings in fruit flies. How would you determine whether a straight-winged fly is heterozygous or homozygous? Well, you can't tell just by looking at it what it's going to be. So what we do is something called a test cross. And this is where you cross the whatever the trait is with a homozygous recessive trait, because this will tell us information. Now here's why. Let's imagine that we have a homozygous dominant fly here, and that would be a straight-winged fly. If we cross it with a curly-winged fly, and we do the Punnett square, we know that all of the children are going to be, well, straight-winged because it's dominant. Now, what if the situation was that we actually had a heterozygous straight-winged fly, and we didn't know this, and we were to cross it with a homozygous recessive or curly-winged fly? What we'll see in their offspring is that half of them actually have the recessive trait. So again, a dominant one is being tested over here, and we should see all straight-winged flies, and the heterozygous one, we should see half straight-winged flies and half curly-winged flies. So a test cross will show us what the genotype of the parent really is. And if you hear lots of noises, that's my dog crawling around on the floor. Number 8. What gamete combinations are possible when the following groups of alleles separate in meiosis? Now remember, you can only give half of your information to your offspring. So when we talk about meiosis, we're talking about forming either a sperm or an egg. And I'm just going to use an egg here because it's simpler to draw. Now we know that we can only give half the information. So we can give either one of the A's. It doesn't really matter which one of those because they're the same, and we can give one of the B's. So the egg will have one A and one B. And in this case, it'll be capital A and a capital B. But it could give the other allele. So could be those ones. Now, if you look at these, capital A, capital B, those are actually the same. So for this first situation, there's really only one possibility. All of the eggs or all of the sperm can only have a big A and a big B. There's just nothing else that could happen. Now, on this next one, it's a little bit different. So we know we can give a big A and a big B like the other one, but there's another possibility here. We could give a big A and a little b. So there are actually two different possible gametes that can be produced from this combination. Now in this next one, we have a totally heterozygous combination, which means we're going to have lots of different gametes. The first one is just like our very first example, a big A and a big B. We could also do the other two, and we'd have little a, little b. Now here's where it starts to get a little tricky. We can do some more crisscrossing. We could get a big A and a little b. And lastly, we could get a little a and a big B. So there are actually 4 combinations for this one. And then the last one, we could have big A, little b, or we could have little a, little b. And those are the only combinations for that. So in summation, there's 1 gamete combination for the first one, 2 for the second, 4 for the third, and 2 again for the fourth. Now I did skip a number in there, so if you notice it jumps from 8 to 10, that's what it says on your handout as well. Black hair is dominant over brown hair in rabbits. Short hair is dominant over long hair. If a homozygous black short-haired rabbit is crossed with a homozygous brown long-haired rabbit, what would be the genotype and phenotypes of the F1 generation? Now first we have to figure out what are the parent genotypes. Now it says homozygous black short-haired. That means that we've got to have 2 capital B's for the black hair and 2 capital H's for the short hair because those are dominant. And we're crossing it with a homozygous brown long-haired rabbit. So these are all little letters because these are all the recessive traits. So if we do this next step very carefully, if we figure out the possible gametes, we're actually going to save ourselves a lot of time because we don't have to set up a big Punnett square for this question. From the first parent, we can only give a capital B and we can only give a capital H, so there's only one possibility, big B, big H. Now the other parent, you can only give a little b and a little h, so the only possibility is little b, little h. So you're about to make the quickest Punnett square of your life. Set up your square. It's literally just a square because this trait goes at the top, this trait goes to the side. All of the kids are going to be heterozygous. 1 big letter and 1 small letter for each trait. And that makes this pretty easy. So the genotype of all the F1 generation is heterozygous, 100%, and the phenotype of all the F1 generation is 100% black and short hair, which is the dominant trait. Now we're going to take these from the F1 generation and cross them with each other, which means it's going to get a little bit more complex. So our parent genotypes this time are 2 heterozygous bunnies. Now we got to do the possible gametes to figure out what we're going to put on our Punnett square. We have 2 big letters, a big B, little h, little b, big H, and little b, little h. And what we do with these combinations is set them on the side. So we'll move these to our Punnett square, and since each parent has the same combination, we put the same letters at the top as well. And you see I've also put them in the same order because what it does is make some patterns for us and helps us see what happens. So now we're ready to fill it out. We bring down the B, bring the other B across, bring down the big H, bring the other H across, and so on. Continue in this manner until you've filled out your Punnett square. Now this can take some time, and practice will make you even faster at this. Okay, so we've got it all filled out. The next thing we need to do is figure out the genotypic ratios. Oh my goodness, there are a lot. In fact, there are 9 different genotypes listed here, and I've written them out. And what we got to do is count how many of each of those genotypes there are. So for big B, big H, there's just 1. So I write 1 down. And then I'll go just down the bottom, and then I look over to the side, and I see there are 2 that are big B, big H, little h. So I write down the 2. And the next one I'm going to look at is big B, big H. And I see just 2. Write it down. Then we'll go big B, big H. Ah, I'm starting to see a pattern here. You notice it's going all the way diagonal, and there are 4 of them there. Right, big B, little h, and there's just 1 of those. The next one's little b, big H. And I think it's just 1 of those. Then big B, little h, 2 of those. Little b, big H, 2 of those. And last but not least are little b, H. There's just 1. Whew, okay, that's done. Our last step is the phenotype. Now for me, I like to have a little shorthand way of making sure that I've counted these correctly. So even when I'm handwriting this, I'll draw little symbols that help me remember what they look like. So here are the 4 possible phenotypes. There can be black short hair, black long hair, brown short hair, or brown long hair. So I'll start with the black short hair ones, and I'm going to use a black circle to remind me these are short haired. And the way we spot those is that any rabbit with a capital B and a capital H is going to have black short hair. So I go through and I find all those possibilities. And I see that it makes a kind of nice triangle, and there are 9. Now I'm going to look for black long hair, and for that I'm going to use an elongated symbol just to remind me that that's what I'm looking for. And I only see 3 of them. It has to have at least a big B and little h's. So there are 3 of those. Next I'm going to look for brown short hair, and I'll use a brown circle for this one. And I need to see little b's and at least 1 capital H. And there are 3 of those. And last are the little b's, little h's, and that makes a brown long-haired bunny. So there is just 1 of those. And this pattern, 9 to 3 to 3 to 1, is consistent. So anytime you have to cross 2 totally heterozygous traits, you will see a 9 to 3 to 3 to 1 phenotypic ratio every time. It's a very common pattern. So if I ask you a question like this, you could answer the phenotypic ratio 9, 3, 3, 1 without even having to do the Punnett square because this pattern is consistent.